So with that, here is Shah's thesis defense, longitudinal patterns of microplastic abundance and potential for food web uptake in the Yakima River.
Thank you, Dr. Rango, and thank you everyone that's here and on Zoom to watch my defense. Okay. So prior to plastic existing, all consumer items were made out of things like turtle shell and ivory, and it was in 1869 that the first plastic product we saw enter the market became available. And this was celluloid, which was a plant-based material, and it was created in 1869 as a result of a contest looking to replace ivory for the game billiards. So we can thank pool for the situation we're currently in. This led to the big synthetics boom between 1900 and 1929, sorry, which is when we saw products like casein and Bakelite and PVC enter the market, and these products are more similar in nature to what we consider plastic today. Plastic became available to consumers outside of the military post-World War II, and it was in the mid-1960s it was by the mid-1960s that plastic had become the most used material in the entire world. In the mid-1970s, researchers working off the coast of Ireland first documented small pieces of plastic in the marine environment, and although they didn't have the term microplastics back then, that's almost surely what they were documenting. In today's world of 2022, plastic is in most, if not all, consumer items, and plastic pollution is everywhere in our world. Imagining a world today without plastic seems fairly impossible, but mainstream plastic production has really only been around for about 70 or so years. In 1950, there were about 2 million tons of plastic produced annually, and that number had skyrocketed to 480 million tons of plastic produced in the year 2020. That means that over half of the plastic ever created has been in the last 22 years. Estimates indicate that by 2060, we're going to see plastic production triple, which means that 1.4 billion tons of plastic will be produced in the year 2060. Due to inadequate waste management, there's about 32 million tons of plastic pollution that enter the environment every year. And to put that in perspective, a lot of us have heard about the Pacific Garbage Patch, and here's an aerial photo of it. It's twice the area of Texas, and based on the estimated weight of the Pacific Garbage Patch, which is 87,000 tons, we can estimate that 366 Pacific Garbage Patches worth of pollution enter the environment every year now. If we continue on the trajectory that we have of plastic production and use and pollution, we can estimate that by 2060, there will be 1,175 garbage patches entering the environment per year. The same properties that make plastic such a useful consumer item are the same properties that make it such a pervasive global issue. Plastic is incredibly inexpensive to manufacture, so it's mass produced, widely distributed, and about 42% of all global plastic is geared towards single-use packaging. So these are things like your salad to-go kits from Safeway, your coffee cups, your water bottles. And once these enter consumer possession, they have an estimated life usage span of 12 minutes, which means that they'll be used for 12 minutes, discarded into the trash, where they will then decompose for about 1,000 years. Plastic is incredibly durable, so unlike organic materials, it doesn't ever biodegrade. It just continues to break down into smaller and smaller pieces. Those pieces are incredibly lightweight, so they can disperse far distances from their point of origin. And aside from being an eyesore and being fairly disgusting, why do we care that plastic is entering the environment at such an incredible rate? There are 4 types of plastic pollution, but for the purposes of my research, I'm focusing on 2 of them. Macroplastics, which are larger than 5 millimeters, such as this glove, break down through environmental degradation like UV light, wind, and wave action into smaller and smaller pieces called microplastics. And there are 2 types of microplastics, Primary, which are specifically manufactured tiny pieces of plastic, like what you'd find in facial exfoliant or toothpaste or glitter. And secondary, which exist as a result of that degradation from larger pieces. The biggest issue with microplastic pollution is that even if we could scoop up all of the garbage patches from all of the oceans and seas around the world, what would be left behind in the aquatic environment are these small pieces, because there's no known way to filter them out once they get there. And once this plastic pollution is in the aquatic environment, it can become bioavailable to organisms like insects, shellfish, and fish, like this fish that was caught in the South Pacific. And what you can see here are macroplastics that were removed from its gastrointestinal tract. And consumption of macroplastics has been known to cause things like blockages, pseudosatiation, which is when an organism feels full but they're not gaining any nutritional value, and that can lead to starvation and death. The larger issue with microplastics is as they break down into these smaller and smaller pieces, they can actually be found in other areas of the body. So what we can see here are the hepatic cells of a fish caught in the Adriatic Sea. And researchers actually documented these microplastics migrating from the gastrointestinal tract between cellular walls and into the cells of the liver. And some of the known negative consequences of these migrating microplastics are inflammation of tissues, which can cause things like sickness and cancer, reduced growth and reproductive success, and chemical leaching and sorption, which is when the additives and chemicals that make up plastic either leach out of the plastic piece into the body and the environment, or they actually pull other toxins from the environment into the body. Research on this microplastic consumption and potential for food web transfer has been well documented in the marine environment, but it's severely lacking in freshwater systems. We of course know that plastic is produced on land, and we've established that we're finding it in marine environments, but how does it get there? Recent estimates indicate that about 80% of plastic debris is transported from land to sea via river pathways, which are actually called plastic conveyor belts by microplastic researchers. Some of the known source inputs of microplastics into rivers are wastewater treatment plants, which discharge treated municipal wastewater directly into rivers, agricultural runoff, so the films and sludges and other plastic equipment used in the agricultural industry wash into fields and orchards, then into canals, and the canals dump directly into rivers. And stormwater drains. So all of the manholes and drains that you pass as you're driving on the road actually drain directly into the river without undergoing any sort of treatment process. We know that microplastics are in rivers, but what we haven't established is how they're distributed in the riverine food web once they get there. And this is what brings me to the objectives of my study. The goal of my research was to describe the potential pathways of microplastics into and within a riverine food web, and to ascertain how microplastic abundance changes along the longitudinal gradient of a river. The crux of my study was the riverine food web, and the base of the riverine food web is biofilm, which is the phylogenetically diverse communities of bacteria, protozoans, and algae that make up that green sludge you see on the river bottom. Another very important energy source in rivers is FPOM, so this is fine particulate organic matter. It's easily transported in the water column, and it's derived from coarse particulate organic matter, or CPOM, which are large leaves and other organic matter that fall into the river, and biofilm. The primary consumers of these energy sources are aquatic insects or macroinvertebrates, and they're so specific in their feeding strategies that they can actually be categorized by these strategies, which is called their functional feeding group. Shredders consume CPOM, collectors filter FPOM out of the water column, scrapers scrape biofilm off the river bottom, and predators consume all three of these functional feeding groups. Aquatic insects are the critical link between the energy source in a river and higher order consumers such as fish. These energy sources are actually susceptible to microplastic pollution. So plastic can adhere to biofilm and be consumed by organisms, and microplastics in suspension in the water column actually highly resemble FPOM and CPOM. So they can be inadvertently consumed by organisms in the water. The food web of a river is incredibly linked to its surroundings. And this is what brings me to the river continuum concept. So my research is grounded in the RCC. Rivers are influenced by the surrounding landscape around them, and those landscapes change as you move along the longitudinal gradient of a river. And insects are the critical link between this changing landscape and what we see in the food web at a specific point in the gradient. Using the RCC allows us to predict changes based on that changing landscape. We can predict food resources, so at the headwaters of a river, we expect more CPOM because it's narrower channels, there's more leaf litter, and very little light penetration. As we extend down the gradient, the channel widens and we have more light penetration, so there's a corresponding increase in biofilm growth. And at the mouth of a river, it's at its widest point. The turbidity increases, the depth increases, we see less CPOM and biofilm, and the primary food resource becomes FPOM. By predicting these food resources, we can predict the proportional abundance of functional feeding groups. And those functional feeding groups can become more vulnerable to microplastics depending on their feeding strategies and where they're located in the gradient. Based on what we know about riverine ecology and some of the preliminary data collected by Dr. Arango, there appears to be two fairly obvious pathways microplastics can take to enter the riverine food web. Plastics floating in suspension in the water column and adhering to biofilm can be consumed by macroinvertebrates, which are then consumed by higher order consumers such as fish. And plastics adhering to the biofilm can be inadvertently consumed by bottom feeding feeders such as this sucker. The field of microplastics is in its infancy. It's really only been around for about 20 or so years, and it wasn't until 2014 at a UN's Global Summit meeting that governments from around the world had a call of action to increase microplastic research. It's because of this infancy that there's a lack of standardization in the field. Every lab collects samples, processes samples, and reports their data differently. And our world is full of microplastics, and this includes you in the audience. Unless you're wearing 100% cotton or merino wool right now, you are wearing plastics, including myself. So it's because of this that it's so easy as a microplastic researcher to contaminate your own samples. And prior to 2020, there were very few published studies dealing with this contamination issue or how to prevent it, mitigate it, or even really addressing it. In preparation for my research, I conducted a pilot study, and I did this in order to standardize my methodology before I went out into the world to collect my thesis samples. And during this process, a contamination issue was discovered, and we linked it back to the equipment and the researchers in the lab. So this pilot study helped me inform my research questions. The first of which was, can lab contamination be reduced and or controlled enough to detect environmental plastic? I also wanted to know, what is the microplastic abundance in the riverine environment, which is the water column, and in the primary energy source, which is biofilm? So I asked, what is the microplastic abundance? Does river geomorphology influence microplastic distribution? And how does microplastic abundance change along the longitudinal gradient of the Yakima River? I also wanted to know if feeding strategies impacted microplastic consumption. So I asked, does microplastic abundance differ among insect functional feeding groups? How does microplastic abundance change along the longitudinal gradient of the Yakima River? And is there a relationship between environmental microplastics and the microplastics we're finding in insects? My study area was comprised of the entire Yakima River. It ranges 214 miles from the headwaters to the mouth, and it's the longest river of origin in Washington state. There are about 400,000 people living in the Yakima River Basin that depend on this river for their agriculture, sustenance, and recreation. And about 40% of the entire employed force in the basin depends on this river. As we can see here, there are distinct zones of usage along the longitudinal gradient of the Yakima. At the headwaters, we have a highly forested area at 33%. And as we near the Kittitas Valley, we can see this shift from forested area to agriculture. And as we near the lower valley, so near the mid-valley and towards the mouth of the river, we can see how agriculturally dominated the basin is at 28% total of land cover in the Yakima Basin. I had 9 sites ranging from Snoqualmie Pass all the way to Kennewick, Washington, and they were roughly equal distance apart at 25 miles. This is my first site located in the Gold Creek tributary. As you can see, it was snowing the day we sampled. This is site 7 located out of Zilla. This is site 8 located in the Thesis Wildlife Refuge. And this is site 9 located just downstream of the West Richland Bridge. All of my sites were located on point bars, and I did this intentionally to standardize flow conditions and to help me answer that second question. Does river geomorphology influence microplastic distribution? I had 5 systematically placed transects that were equal distance apart, ranging downstream to upstream. And at a randomized point between 0.1 meters and 0.5 meters depth, I collected a water column and biofilm sample. I also chose the 4 most representative macrohabitats at each point bar, and I collected a macroinvertebrate sample at these points. I compiled those 4 samples to form one composite sample, and that allowed me to have a general idea of what the macroinvertebrate community looked like at each site. When I was preparing and doing all my literature review and trying to come up with my study design, I came across most studies focusing on surface water sampling. So this is when you take a grab sample from just the surface of the water column, and that is inherently biased. Because of the different chemicals and additives that make up different types of plastic, all plastics have different densities and buoyancies, and if you only sample the surface water, you're biasing your samples towards more buoyant plastic. So it was very important from the beginning for me to get an entire snapshot of what the water column looked like at that moment in time. So to do this, I collected 4 1-liter samples from the surface, 25% down, 50% down, and the river benthos. And I compiled these into one composite sample that provided me with that snapshot in time. My biofilm samples were collected by scraping the surface of each randomly selected rock or sediment sample from each transect, and standardizing all of those samples to centimeters squared so I could compare microplastics per area between sites. My macroinvertebrate samples were collected using a D-frame kick net, and I fixed each sample in 70% ethanol in the field, and those samples were then transported back to the lab and identified to family. Once my samples were collected, I brought them back to the lab, and this is where I began the process of chemically digesting them. All of my water column samples were sieved through a 250 micron sieve, and this was done to condense that 4-liter volume to a more manageable sample. My biofilm samples were poured directly into beakers, and the water column and biofilm samples underwent the wet peroxide digestion method. So this is where we chemically digest all organic material, and it takes between 24 to 36 hours per sample. Once my insects were identified to family, I carefully washed each individual insect with deionized water, picked off any external plastics, eviscerated each insect, and then digested them in hydrogen peroxide for 72 hours. And what you can see here, the arrow is indicating a blue microfiber that was adhered to the inner abdominal wall of this immature stonefly. Once all my samples were chemically digested, They were each filtered through a Buckner funnel and onto a gridded Petri dish. Those Petri dishes were then prepared for enumeration. So using a dissecting scope, I analyzed each Petri dish at 2.5 to 3 times magnification for the water column and biofilm, and 4 to 4.5 times magnification for the macrovertebrate samples. Each plastic was categorized as either fiber or fragment, and I utilized conservative counting. So if I wasn't 100% sure that what I was looking at was a plastic, it wasn't counted towards my final analysis. Oh, sorry. What you see here is an immature stonefly prior to digestion, and in its mouth is a bundle of organic materials. And it's difficult to see, so I've highlighted it in red, and embedded in those fibers are red microfibers. So this implies that aquatic insects are inadvertently consuming microfibers because they blend in with their environment sources. The first question I wanted to answer is, can lab contamination be reduced and or controlled enough to detect environmental plastic? To do this, I conducted a contamination mitigation experiment. I wanted to know, does protective gear help mitigate contamination from researchers? So I took 20 samples from the Yakima River, and I processed 10 of them wearing protective gear, which was a very cheap poncho. And then I processed 10 of them without that protective gear or poncho, but while wearing a long sleeve, bright orange fluorescent orange shirt, so that I could follow these very distinct microfibers all the way through the procedure. I also wanted to know, we know we have to have controls, that's just kind of obvious, but how well do they work? So at the end of my entire study, I compared all of my control samples to my environmental samples to see how they differed. To analyze this data, I used a one-way ANOVA to compare the microplastics per sample when I wore protective gear versus no protective gear. And to compare my control to environmental samples, I used a paired t-test to compare fibers per liter in the water column. And fragments per liter, I used a Wilcoxon sine ring test because of non-normal data. To compare the control versus environmental biofilm data, I used a Wilcoxon sine ring test. And when I looked at my control versus environmental insect samples, I used a Mann-Whitney U test due to unequal sample size. The answer to my first question, does protective gear work? I found that there were significantly more microplastics when I didn't wear a poncho compared to when I did. When I compared my controls to my environmental water column data, you'll see fibers on the left and fragments on the right. I found that there were significantly more microplastics in the environmental samples in the water column than the controls. When I looked at the biofilm data, again you'll see fibers on the left, fragments on the right. I found that once again, there were significantly more microplastics in the environmental biofilm samples than in the controls. And finally, when I compared my macrovertebrate samples, I found that there were, again, significantly more microplastics in the environmental samples compared to the controls. So what does all of that mean? It means that controls can be used to quantify exogenous plastic, and that we can actually quantify this background contamination noise that our world and researchers can cause in their labs. So this gives us more confidence in the amount of microplastics we're measuring from the environment. All of these results ended up in me developing new protocols. So I found that protective gear works. In my lab, you could not enter my lab, it was a locked door policy, unless you were wearing this goofy suit. And I know it's made out of plastic, it's ironic, but what we were worried about was fibrous contamination. So this helped mitigate that contamination. And I also couldn't afford like $1,500 hazmat suits, so. I took a cleaning methodology that was developed by leading researchers in the field, and I further expanded upon that by implementing a pressurized water system in my lab to completely clean all of my gear. I took a method that's used by leading researchers, Kirsten Skalska, Rockman, Holing, all these big names in this field, and I implemented it. So we quantify environmental samples with controls by color and morphology. So what does that mean? If we have 3 red fibers in our controls from a site, I subtract 3 red fibers from my environmental samples from that site. And that's what quiets that noise so we can see what those environmental samples actually look like. Implementing all of these methods and these study designs gave me confidence in my design, in my methodology, and in the results that I collected. And using this methodology resulted in a total of 354 samples for my entire project. When I looked at my water column data, I found that 93% of my environmental water column samples contained plastic. So every single site had plastic in the water column. To analyze this data, I used a paired t-test to compare fibers per liter to fragments per liter. And to deduce if there were any longitudinal trends or differences among sites, I used a general linear model. So I estimated the microplastics, fibers, and fragments as a function of the explanatory variables site, transect, velocity, and depth. When I looked at what the microplastic abundance in the water column was, I found that there were significantly more fibers in the water column than fragments across the entire Yakima River. When I investigated the question, does river geomorphology influence microplastic distribution, I found that for fibers per liter, which is here on the y-axis, and transect on the x-axis, excuse me, there were significantly more fibers per liter at transects 3 and 4 compared to 5, 2, and 1. And we'll go into what that might mean a little later on, but this is where those transects are located on our point bar. When I looked at fragments per liter, again on the y-axis and transect on the x, there was no significance. So even though they were both water column samples, the different types of plastic didn't share those trends. When I looked at how microplastic abundance changes along the longitudinal gradient of the Yakima River, you'll see microplastics per liter on the y-axis and site on the x-axis. I didn't see any longitudinal trends, and I also didn't see any differences among sites. So fibers are pink and fragments are blue, and they are relatively homogeneous across the entire Yakima, so no one site showed a significant difference. When I examined my biofilm samples, I found that 96% of my biofilm samples contained microplastics. So every site had biofilm with microplastics in it. To analyze this data, I compared fibers per centimeter squared to fragments per centimeter squared using a Wilcoxon sine ring test. I looked at any longitudinal trends or differences among sites using a generalized least square model. And I estimated microplastics as a function of the explanatory variables site, transect, velocity, and depth. What I found when I looked at the abundance of microplastics in the biofilm is that once again, there were significantly more fibers found in the biofilm across the Yakima River than fragments. When I investigated river geomorphology, I didn't see any differences. So you'll see fibers per area on the y and transect on the x. And fibers in the biofilm didn't share those same patterns that fibers in the water column shared. When I looked at fragments per area, I also didn't see any significance. So it appears that river geomorphology doesn't influence the distribution of microplastics in the biofilm. When I looked at how microplastic abundance changes along the longitudinal gradient of the Yakima River, we have microplastics per centimeter squared on the y-axis and site on the x-axis. Again, fibers are pink and fragments are blue. When we looked at our fibers, I found that site 8, which is located just at the end, had significantly more fibers per centimeter squared than site 7, or than 4 other sites. When I looked at fragments per centimeter squared, I found that once again, site 8 had significantly more fragments per centimeter squared than site 6. And site 3, which is just here, had significantly more fragments per, fragments per centimeter squared than 7 other sites. When I looked at my macroinvertebrate data, I found that 68% of my samples contained plastics, which means that all sites, except for site 8, had at least one insect with microplastics in its gut. And site 8 wasn't an anomaly because we found insects without plastic, but because we didn't collect a single insect. So it wasn't counted in my final analysis. To analyze this data, I looked at any longitudinal trends or differences among sites using a generalized linear model with a gamma distribution. And I estimated microplastics as a function of the explanatory variables, functional feeding group, and site. I compared the amount of fibers versus fragments found in insects using a Wilcoxon sign rank test. And I wanted to know how microplastics in the environment compared to what we're finding internally in insects. So I used a Kendall's rank correlation to do this. You'll see fibers per insect on the y-axis and site on the x-axis. And when I looked at whether or not there were any longitudinal trends in microplastic fibers per insect, I did find significance at site 7. So it was significantly different from all other sites. But fortunately, or unfortunately, depending on how you look at it, when I dove into why this might be, I found that it was because of a contamination issue. So one of my controls from this site had an unusually and impossibly large number of microplastics in it. And when I removed that control, that significance of that site went away, which further underscores the need for implementing controls in this research. I did find that site 3 had significantly more fibers per insect than site 2. When I looked at microplastic fragments in insects and how they change along the Yakima River, I didn't find any significance. So it appears that across the entire Yakima River, microplastic fragments in insects are relatively homogeneous. I wanted to look at how the fiber consumption in insects differed based on their functional feeding group. So you'll see fibers per insect on the y-axis and family on the x, which is how we get to functional feeding group. And there was no significance. So it didn't appear that feeding strategy had anything to do with the amount of fibers consumed by these insects. And when I analyzed fragments per insect, I found that once again, there was no difference between the amount of fragments consumed and what their functional feeding group was. So it appears that for the insects in the Yakima Valley, they don't really care what their feeding strategy is, they're going to eat plastic. And this was the most surprising. So you'll see microplastics per insect on the y-axis and fibers and fragments on the x-axis. If we think back to our water column data and biofilm data, fibers were the most prevalent microplastic in their environment and in the primary food source. So I figured, well duh, they're going to have more fibers internally because it's more prevalent. But no, I found that there was no statistical difference between the amount of fibers or fragments consumed by insects. When I went to look at the relationship between environmental microplastic and plastics in insects, I conducted 16 Kendall's rank correlation tests to figure out if there was any sort of correlation. No, there is no correlation between the amount of plastics in the environment of the Yakima River and the plastics we found in insects. I know I just threw an incredible amount of graphs and figures and p-values at you, but what does all of that mean? I found an average of 3.4 microplastics per liter in the Yakima Valley, in the Yakima River. And I wanted to compare that number to 3 other similarly used rivers in the United States. And I found that while our number of microplastics per liter is slightly elevated, it's still comparable to these rivers. And more importantly, we've established that we have a baseline amount of microplastics. So now our river can be grouped in with these other rivers and be studied by other researchers. I found an average of 0.52 microplastics per centimeter squared. And unfortunately, to my knowledge, no one else has done this. I haven't been able to find any other comparable studies to compare those numbers to. Typically in microplastic research, when researchers are looking at the relationship between biofilm and microplastics, they're actually looking at biofilm growth on microplastics, not the other way around. So while I wasn't able to compare this value to anything, I did show that there's a prevalence of microplastics in key food resources in the Yakima River. And those numbers probably don't mean anything to you. So to apply those numbers to real life, I think we've all spent, hopefully all of us have spent some time in the Yakima River Canyon. And this picture is Site 5. So this is located just upstream of the Mile 8 boat launch. And what these numbers extrapolate to is that if you were to stand on this bank in May of 2021, for every second that passes in the water column, there are 300,000 microplastics passing by you. That means that for that site, which was a 60 meter by 60 meter plot, there's about 16 million microplastics on the river bottom just at that one location in the Yakima River. I also found that fibers are most prevalent, except in insects, and this does not deviate from what other researchers have found. Fibers appear to be the most prevalent microplastic type, and this further underscores wastewater treatment plants as that point source of microplastics into rivers, as that's the most dominant microplastic type in the effluent that's discharged into rivers. When I went into this study, and Dr. Rango can tell you, I kind of thought it was a shoo-in. I thought, well, we're starting from a forested area. You move through all these cities, that's a cumulative human impact. There's got to be more microplastics at Site 9 than Site 1, and we'll see a nice, even gradient as a linear increase. I didn't find that. When I dove into why this might be, I found that some studies find nice linear trends in microplastic abundance, and some studies don't. Some studies have no consistency, no patterns, and I found that in microplastic research, inconsistency is consistent. I did find differences though. So while I didn't find differences among sites in the water column, I did when we looked at the biofilm samples. So when we consider fibers per centimeter squared, Site 8 had significantly more fibers than 4 other sites. And when I looked at the fragments per centimeter squared, I found that Site 8, once again, had more fragments per centimeter squared than one other site. Site 8 is really important, or is really interesting to look at. It's about half a mile downstream from the confluence of multiple agricultural inputs. And when we consider how agriculturally dominated this lower valley is, all of those canals that are across the entire lower valley drain into this one point. So this might be what we're seeing here. It was also really important, if you notice, sand. This was my only site that I sampled in the Yakima River that had sand and no rocks or cobble or any sort of other type of sediment. When we look at Site 3, it had significantly more fragments per centimeter squared than 7 other sites. And this site is interesting because it's about 100 meters downstream of a very frequently used boat launch. Boats are made out of plastic, and this boat launch is used year-round, so boats are put in and taken out. People congregate here to tie flies, clip ties, and get ready for fishing expeditions. So it's possible that we're seeing an increase in microfragments at this site because of recreation. This is my Site 3, just so you can see it, lots of cobble. And while I didn't have any statistical significance to this site, I wanted to talk about it. So Site 1 was my first site. It's located in the Gold Creek tributary. Again, I thought, highly forested area, very little upstream human population. It should have the fewest amount of microplastics. But what I found is that Site 1 had the second highest number of raw microplastic fibers in the entire river. When I looked into why this might be, I found that there is a very well-established link between atmospheric deposition of microplastics and how many microplastics we're finding, specifically fibers. So fibers can actually be transported in the atmosphere and deposited in precipitation. Site 1 has the highest amount of precipitation in the entire Yakima River. Additionally, Site 1 is about an hour from Seattle, and the Gold Creek Pond area is inundated year-round by recreators. If you can think of any sort of recreation activity, it's done at Gold Creek year-round. And there is well-established links between recreation and synthetic fiber abundance in the environment. So what this highlights is that maybe there are different input sources of microplastics into the environment than we previously thought. And these differences that I found at these 3 different sites might be attributed to plastic deposition. When we think of rivers, we assume that, well, something drops into the river upstream, it carries it downstream, and that's it. But that's not true. Researchers like Tim Homeline are establishing that rivers can actually act as storage for pollutants and other foreign products, and more research needs to go into uncovering this deposition. When I looked at the geomorphological influence of river geomorphology and microplastic deposition, I found that for the water column, fibers per liter showed significance among transects, but fragments didn't. And when we consider the different additives and chemicals in plastics and their different morphologies, it kind of makes sense. Fibers are lighter, they're buoyant, and they're subject to suspension and resuspension in the water column. But fragments are dense, and they tumble along the river bottom, and usually they get entrenched in sediment. So what I think we've found here is that fluvial geomorphology might be affecting microplastic fiber distribution in the water column. And because this is the most prevalent microplastic type, we need to dedicate more research to how rivers are changing or affecting this deposition. I was so stumped by the fact that there were more fibers in the environment, but not in insects. I couldn't find anything in the literature. And what I think we have found is that maybe insects, in the Yakima at least, are preferring fragments over fibers. That hasn't been documented yet, so I think that this portion of my study might be one of the most important, because we're showing that feeding strategies and prevalence of microplastic type in the environment might not mean as much as we thought. I didn't find any differences in plastic ingestion by functional feeding group, and this directly contradicted other studies. So there are very few researchers that specialize in microplastics and macroinvertebrates, but one of the leading ones, Windsor out of the UK, has well established that collectors who filter microplastics out of the water column have fewer microplastics in their guts, and scrapers, which scrape the biofilm, have more. And I didn't find that. So what I think we've established in the Yakima is that functional feeding groups are equally susceptible to ingestion no matter what their strategy is. No matter what, the end result of this data indicates that we found plastics in insects, and that implies that plastics can move into higher trophic levels. And trophic transfer of microplastics into apex predators has been documented. It's been seen in insectivorous Eurasian dippers. So more research into how this trophic transfer occurs is very much needed in the Yakima River. So why does any of this matter? To the best of my knowledge, my study is the first of its kind. I have not come across another study that has looked at an entire longitudinal gradient of a river and analyzed microplastics in the water column and the biofilm, and not just one insect, but 4 different functional feeding groups. We've kind of discussed contamination in this talk. My study employed 7 out of 10 recommended protocols for mitigating contamination, and the average that other studies follow is 4 out of 10. I didn't have a huge budget for this study, so I'm hoping that my research can act as a framework for other low-budget studies so we can begin to collect microplastic research, compare it, and extrapolate that data so we can have an idea of how bad this problem actually is. I'm really hoping that habitat managers in the Yakima Basin use this data. The Yakima River is Washington's only blue ribbon stream, which is a category reserved for the utmost pristine fisheries. And I think what I've documented here is that we need to be focusing on other sources of microplastic pollution in rivers. Most studies focus on wastewater treatment plants and urbanization, but for areas that are agriculturally and recreationally dominated, like the Yakima River Basin, we need to be looking at what other sources there are. And finally, the bioaccumulation of toxins. So as this sorption and leaching of chemicals happens with plastics, we're documenting plastics in insects. Insects are consumed by many other species, bats, fish, birds. Basically, they're the link. And what we could see in the future is a bioaccumulation of these toxins into the food chain, which ultimately leads up to us. I always like to be transparent, and I would like to put some considerations on the table with the results of my study. I used visual analysis. So this is literally counting every single petri dish, 354 of them, under a scope and manually counting each plastic. This is not as powerful as spectroscopy. So methods like FTIR and mass spectroscopy are more accurate, and it's possible that my results may not align directly with what those results would find. I used a sieve to funnel my water column samples. So it's possible that because I used a sieve for my water column but not my biofilm and insect samples, maybe I counted fewer plastics in the water column than in the other samples, and that's why we didn't see that correlation. And finally, temporal flooding may have obscured any spatial patterns that do exist in the Yakima. I was sampling in May of 2021 between flood pulses, and it's possible that those flooding events diluted the water column, so I counted fewer. It's possible that those flood pulses flushed the sediment. And it's really important to recognize that rivers flow year-round. I sampled in May of 2021, so this study may not be representative of patterns that are seen at other times in the year. I think that future research should be dedicated towards replicated longitudinal studies. So we need to be comparing watersheds to each other and similar use rivers to each other. We need to replicate temporal studies. If I was a masochist and wanted to stay in grad school forever and had unlimited funding, I would have loved to do the study in January, May, August, October, so that I could compare how those patterns fluctuate at the same point at different times of year. And we need to dedicate more energy to geomorphological specific studies. So we've established that rivers are not just transport mechanisms. They can actually store pollutants, and we need to look into this to see how it's affecting our rivers. I would really like to acknowledge a few people. First and foremost is Dr. Clay Arango. The last 2 years have not been easy, and I don't think I've told you thank you enough for keeping me on track and making sure that I have the most powerful study I possibly could. My committee, Dr. Jason Irwin and Dr. Paul James, my field assistants, David Revel, Will Raymond, and Josh Goble, could not have done this without you. Will spent 8 to 10 hours for weeks on end listening to true crime podcasts and didn't complain once. My family, friends, and grad cohort, of course, thank you. And I'd really like to thank the funding sources I received that allowed me to conduct my research and survive during grad school. And I'd really like to thank you guys for taking the time to listen to my defense.
Thank you. At this point, we'll take any questions from the audience and also from Zoom. So Mark, if you could